As you know, today we are going to discuss a new chapter, blood banking, or the update, the full name for that is immunohematology and transfusion medicine. This is the correct full name for these chapters. And as the name implies, there are two departments, two classes in these chapters. The study of immunologic responses to the blood components is called immunohematology, and the medical practice and techniques associated with procurement, processing, and distributing the blood and blood components to patient is known as transfusion medicines. There are two main categories, two main departments in these chapters. Both of them are associated with a blood transfusions, and we call a blood banking or immunohematology and transfusion medicine. Now, okay, there are regulations about blood banking, about blood transfusion. It is really, really important. And as you see, FDA and Center for Medicare and Medicaid Service, Occupational Safety and Health Administration, OSHA, and State Department of Health are involved in such regulation. Okay, now the benefits and reason for transfusions consist of restoring or maintaining the oxygen carrying capacity. If the oxygen carrying capacity is too low, you have to maintain, you have to restore it, to add erythrocytes, okay, to transfuse erythrocytes, because oxygen carry via hemoglobin, and hemoglobin carry via erythrocytes. Now, the other reason is that restoring or maintaining blood volume, the total blood volume is important to maintain the blood pressure. Okay, if you lose a blood, what that means, the blood volume will decrease, the pressure, the blood pressure will also decrease. Okay, and sometimes you need to replace coagulation factors. There are some coagulation factor deficiencies, genetic or acquired coagulation factor deficiencies that you need to replace them. And restoring or maintaining leukocyte functions. Okay, in some cases, such as HIV or some kind of leukemia after chemotherapy or radiotherapy, the function of the leukocytes is changed. If they become dysfunctional leukocytes, or the number of leukocytes may be decreased, and the leukocytes are not able to defend the body, and you have to add some leukocytes in order to strengthen the defense of the body. Now, all these reasons show us that in such cases we need to transfuse either whole blood or packed red blood cells or plasma. It depends on the need of the bodies of the patients. If there is a okay, I mean, there is a low blood volume, okay, and you have to transfuse whole blood, okay, which whole blood contain formed elements such as red blood cells, white blood cells, and platelet and plasma. Okay, if the packed red blood cells have essentially replaced whole blood in transfusion. Okay, if only the oxygen carrying capacity of the body is decreased, the number of the red blood cells is too low, or the concentration of hemoglobin is too low, you can transfuse only red blood cells, which we call packed red blood cells. Okay. Irradiated red blood cells is in some cases important because you will desynthesize them. Okay, they are not able to react with the patient's antibodies, okay, in order to avoid graft versus host disease. Okay, you will okay, by irradiation, okay, you will prevent a immunologic reactions. Okay, plasma is a liquid part of the whole bloods. Okay, whole blood minus cell components such as white blood cells. There are different versions of the plasma that we are using. These are fresh frozen plasma and plasma derivatives, which such as factor 8 or cryoprecipitate, and plasma substitutes. Okay, which kind of plasma that you should use to transfuse to the patient is based on a need of the patients. Okay, if there is a coagulation factor deficiency, like you can only give a plasma derivative which contain such factors. Okay, now let's talk about the fresh frozen plasma, which is a most common version of the plasma that we are using. This is a source of all coagulation factors. As the name implies, it's a freshened frozen plasma which protect the all coagulation factors, and is a source of heat liable coagulation factors. Okay, because this is a fresh frozen plasma, and you also protect the heat liable coagulation factors, and good for treating immune deficiency because, as you know, the antibodies included in plasma. Okay, if you use the fresh frozen plasma, that means you can provide the all coagulation factors and antibodies to the patient. Okay, platelets are other a kind of cell component of the blood to prepare to obtain plasma from the whole blood we call platelet-pheresis. Platelet-pheresis means to obtain platelets from the whole bloods. Okay, there are devices. Okay, you take the whole blood of the patients from its vessels into such devices, and these devices will separate only the platelets, and the rest of the blood will come back into the patient's circulation. Okay, such as cool system that we are using to obtain platelets only. Okay. Okay, the random donor platelets concentrates, the other, that we are using. For that, you must be monitored for bacterial contamination. Okay, because you are now obtain such platelets manually. Okay, and it is probably that you may contaminate them. Okay, and we also store such kind of platelets at 24 Celsius degree at the room temperature in order to save them, to not harm the platelets. The optimal storage temperature for platelet is room temperature, but that means it has a risk of bacterial contamination at the room temperature, and you have to monitor for bacterial contamination always. Okay, and these kind of platelets are useful in case of massive blood loss and replacements. And also they can stimulate production of HLA antibodies. Now, the donation of blood, selection of donor, collection of the whole or component of blood, storage them and processing them are all really important. And we need to take a group of precautions and follow several rules. And the donor selection and identification rules are under these links. Okay, and we need to update ourselves. We need to take a look whenever we need to collect a blood and process them. Okay, there are donor deferral criteria. The people that you shouldn't obtain blood from that. And these donor deferral criteria may update by the time, and you have to take a look each time when you need to obtain a blood or any kind of the blood components. Okay, you can either obtain a whole blood, okay, or red blood cells, and you also need to use anticoagulants and preservatives and labeling is important. As I said, storage of the blood and processing are really important, and we have to know the procedure for donor referral criteria for collection, storage, and labeling and processing of whole blood or but a kind of blood components. Now, in terms of the blood processing, there are several steps that we need to follow, and routine blood screening tests are most important steps in terms of blood processing. And you have to test the blood of the donor for syphilis, hepatitis, HIV, okay, or human T-cell lymphocyte viruses, West Nile, or Trypanosoma cruzi, and some other microorganisms. And to be sure that the donor blood is free from such viruses or microorganisms. Now, as you see here, for Zika viruses, for COVID-19, Ebola, and also you have to fill out the questionnaire, okay, because sometimes the donor educational material, the donor history questionnaire give us some information that we should refer the donors, okay? Let's say if a patient came from an area of the malaria endemics area, and you shouldn't take obtain a blood from such patients. Okay, in some case, patient may be a healthy, okay. However, the donor history questionnaire tell us that he came from a area of endemic plasma. I mean, the malaria endemic areas, okay, or such other issues. Now, okay, type of the blood donation, there are two main ways that we obtain the blood. These are autologous transfusion and homologous transfusions. In the autologous transfusions, which is the safest blood a recipient can receive, receive his or her own blood, okay, which prevent transfusion-transmitted infection disease and eliminate the formation of antibody to antigens, right? Okay, your red blood cells, the safest for you guys. Okay, and in transfused red blood cells from others, and the possibility of the graft versus host disease, okay, will prevent via autologous transfusions. Okay, in the homologous or direct transfusion, the patient directly solicits blood from for transfusion from family or friends, which carry some risks of transfusion-transmitted infection disease or graft versus host disease. Okay, now in order to obtain blood and to transfuse such blood to the patients, you also need to perform some tests, okay? The cross-match test, right? In order to see there is no reaction between donor and recipient bloods. Okay, and for that, you have to prepare antiserum, okay, means of detecting antigen antibody reaction. You need to obtain antiserums, okay, in order to perform agglutination tests, in order to monitor hemolysis, okay. Now, preparation of antisera requirements, reaction of antisera with red cells are seen in processes of cross-matching. Now I am going to explain each of them step by step. Okay, first, as I said, we need antiserums. There are two ways to obtain antiserums to use for cross-matching. The first one is monoclonal antisera are produced by hybridization, which is a DNA technology that we are using, a fusion of a single clone of human neoplastic antibody producing cells with synthesized splenic lymphocytes, which is obtained from a rodent species, and you will be able to use a segment of the DNA which produce a specific antibodies, okay? And as you see, these antiseras, okay, monoclonal antibodies can be either monoclonal or polyclonal, okay? Polyclonal antisera, okay, they have as you see, they are able to recognize multiple epitopes. However, monoclonal, they are able to recognize a single epitope. There, as you see on the, this is a segment on the Fab, which we call Fab, part of the antibodies. Okay, it has the antisera that we produce only is recognize only one epitopes. Other is able to recognize multiple epitopes. Okay, now this is the one way that we produce antiserums, anti-A, anti-B, and anti-D. Okay, will recognize group A blood groups. This will recognize group B, and this is will recognize Rh positive blood groups. Now, the second way to produce antisera is you are going to inoculate antigen of desired to the animals, okay, and this animal will produce antibodies to the antigen that you inoculated, and then later you are going to take the blood of the animals and then purify antibodies in order to prepare antisera. Okay, these antisera you are going to use for cross-matching. Now, the requirements of antisera are important and regulated by the institutions that I show you. It must be specified for the antigen to be detected. Okay, let's say anti-A should be specific to the A antigen, right? it must have a sufficient concentration or titer to detect antigens. Okay, what does means if you have a low level of antigen in the sample, it should detect these low levels. Okay, okay, which it should be able to detect a low level of the antigens. Okay, antiserum must have a certain avidity for or strength of reaction with corresponding red blood cell antigens. Okay. Now, when the antiserum is, or we prepare the serum, okay, via recombinant DNA technology or via animals, and then now reaction of antisera with the red cells, which is we are using in a cross matchings. Okay, when antisera mix it with red blood cells, an antigen antibody reaction may or may not occur. Okay, this is which we are using as a control in the cross matchings. If a reaction does occur, okay, the corresponding antigen must be present on the red cells, and the result is positive. Okay, you are going to use, okay, let's see, antisera A, and then add onto the red blood cell of the patient. If the reaction, okay, is occur, you see a positive blood group. Okay, now if the reaction does not occur, the antigen is absent and the result is negative. For example, a positive reaction with anti-A, antiserum, demonstrate the presence of the A antigen on the red blood cells. What does means the group A, only the group A blood, the erythrocyte of the group A blood carry A antigen, and they will react with anti-A antiserum. If the patient sample is positive with anti-A, that means this is a group A blood group. Okay. Now, how we can see whether it's positive or negative? Okay, it is about the agglutinations. Okay, agglutination is clumping of red blood cells caused by reaction of a specific antibody and antigen on the red cells. Okay, you are adding antiserum, which include antibody, to the patient's erythrocytes, to the donor erythrocytes, or if there is a antigen on the red blood cells specific to the antibody, they will react. This reaction, to form a antibody antigen complex, result in agglutination of the complex. Now, the positive antigen antibody reaction result in immediate combination of antibody and antigen on the red blood cells, and followed by the visible agglutination, okay, which takes longer to form. You have to wait. As you see here, now you add antibody to the patient's antigen, red blood cells, and then this is a how they look like. Now, there is no agglutinations here. Okay, no agglutinations. Negative, that means, and these are positive tests, okay, for a certain blood groups, certain antisera. However, the strength of the positivity is different. It can be 1 positive, 2 positive, or 4 positive. Okay, now anti-human globulin reaction, okay, as we see here now, anti-human globulin antibodies form a bridge between adjacent erythrocytes synthesized with human immunoglobulins. Now, as you see here, now these are human immunoglobulin G blood groups antibodies. Okay, this is antiserum, and then you put this antiserum on the erythrocytes, and if these erythrocytes carry an antigen specific to these antibodies, they will attach to each other. Okay. However, it might not enough to see a visible agglutination. Now these are synthesized erythrocytes. Okay. In order to make a visible agglutination, now you add anti-human immunoglobulins. These are antibody, okay, to the antibodies, antibodies, okay, because these antibodies, which has specificity for the FC portion of the human immunoglobulins, these portions, okay, here. As you see, these are anti-human globulin G, and they are specific to the FC part of the antibodies, and then which result in a visible agglutinations. Okay, these are a appearance of the agglutinations. No agglutinations, as you see, dark, turbid, homogeneous, macroscopic appearance. And then here you see a tiny, okay, many tiny agglutinations, 1 positive, okay, 2 positive, 3 and 4 positive agglutinations, a more stronger one. Okay, now we already have a antisera, anti-hemoglobin serums, and patients' red blood cells. Which techniques that we are using to specify, to test the blood groups. The traditional the specimen of choice for all blood bank testing has become plasma. The sample, the traditional sample, is plasma, and there are different kind of the techniques that we are using to obtain the blood groups, to test the blood groups. Gel technology, microplate testing methods, or solid phase red blood cell adherence methods that we are using to obtain to identify the blood groups. Now you see here the gel or column agglutination technology, how they look like, the 4 positive, 3 positive, 4 positive agglutinations, okay, and this is negative, okay, and this is an example for a solid phase adherence technology. And you see here antigen-coated valve. Okay. You add a serum and low ionic strength solution to get into these valves, incubate them, and then wash out. And now some antibodies which should be supposed to be in the serum. If you have antibodies specific to they will attach to these valves, to this. This is antigen-coded valves, If you have a antibody specific to these antigens, they will attach to these antigens, And then you will wash them and add indicators, red blood cells, And now centrifuge them, and then you will, look at to the valves, the top view of the each valve, strong positive, as you see here, the erythrocytes, the indicator red blood cells will attach all these antigens, and weak positive and negative test. Now that means we also need to discuss the ABO red blood cells groups. ABO phenotypes, genotypes, ABO typing procedure, and these are front typing, reverse typing, red cell typing for antigen, and others. Now, you may know from biology 1240, the blood groups that we discussed before, the ABO phenotypes determined by direct testing, and they consist of the groups. The ABO system consists of groups or phenotypes, and these are A, B, AB, or O, right? And these four groups may be explained by the presence or absence of two antigens on the red blood cells. And these are antigen A and antigen B. If a person belongs to group A, that means their erythrocyte of the patients carry A antigen. A group B person have B antigen on their red blood cells. Group AB person have both A and B antigen on the surface of the red blood cells, and group O people have neither A neither B antigen on their red blood cells surface. Genotypes is different from phenotypes, right? And gene on the chromosome 1 determine the antigen present on the red blood cells. The gene 1, we have 23 chromosomes. Chromosome 1 carry a gene which determine the blood groups, and 3 allele genes can be inherited in the ABO blood system, and these are allele A, B, and O. Usually there are 2 alleles for each character, each traits in our body, but for some traits, some characters, we carry more than 2 alleles, and we call multi-allele genes. Okay, in terms of the blood, ABO, red blood cell groups, there are 3 alleles. They determine the blood groups. Okay, because each person has 2 genes for any traits, but we have 3 alleles. Okay, the power. Okay, what does means 3 power of 2, right? The combination of these 3 alleles to make a 2 genes, okay, can result in different combinations. Possible combination of alleles are AA, okay, both of them can be A or one, A1O, AB, BB, BO, or both of them, both gene can be O, okay? What does means these 2 phenotypically group A, okay, group AB, group B, right? And need to here, these 2 phenotypically are group B and group O. Okay, for group A blood group and group B blood group, there are 2 type of genotypes, 2 genotypes. Okay, they can be either both AA or AO. Okay, since the O gene is recessive, okay, and the appearance, the phenotype will be A, okay? Now, what determine these antigenic features? Okay, there are 3 antigens, A, B, and H. The rest of the structure almost the same. However, if the immunodominant sugar is L-fructose, and this is okay, if the immunodominant sugar is N-acetylgalactosamine, and then it's called A antigen, and if the immunodeterminant D-galactose, as we see here, this is known as A B antigens, and these are the antigenic features of the blood groups. Okay, now it is your turn to send your responses to these questions. Okay, let's go ahead and send your responses. Are you there? Are you awake? Okay, let's see. Okay, one of our classmates, they usually only join to the Zoom, but is not here. Okay, now see. Okay, all the following are benefit of the reasons for blood transfusion except okay, the first 3 are the benefits of the transfusions. Okay, replace antibiotics in treatments of the patients with the granulocytic infections is not a benefit of that. Replacing of the leukocytes maybe, but replacing the antibiotics is not, okay? Next one, fresh frozen plasma is characterized by all of the following statements: is a source of heat-liable coagulation factor, a source of all coagulation factor, transported with dry ice to keep frozen, good for treating immune deficiency. Okay, which is not correct among these options? How we transport and store the plasma, fresh frozen plasma. Okay, let's see. Okay, the correct answer is C. Okay, the transported with dry ice is not correct. Okay, the others is okay. Following test is required in the screening of potential blood donors for transfusion-transmitted disease: hepatitis B surface antigen, hepatitis C antibody, hepatitis B core antibody, all above. One more. Thank you. Okay, it says all above. Let's see the, okay, show the correct answer. Can you see the tick here? Is all above. Okay, there are several tests that we need to perform, okay, in order to save our patients, to protect our patients, right? Okay, no, let's go ahead. Okay, we already talked about the blood, the ABO blood systems, okay, antigens, antibodies, and you may know these slides from biology 12, 14, okay, maybe from other classes, I'm not sure. However, your genotype for gene 1, okay, for chromosome 1 determine the type of antigen present on your blood cells, as we see here, okay, which is your blood types, okay? If your erythrocytes carry a antigen, your blood types is group A. If they carry B antigen, your blood group will be B. If they carry both antigens, okay, these are the location of the antigens, the surface of the red blood cells. Okay, in order to detect antigens, you have to use red blood cells, right? Because antigens are located on the surface of the red blood cells. Okay, in order to determine antibodies, you have to use plasma, serum. Okay, because antibody present in the serum, in the plasma. Okay, and now antibodies against other blood antigens. Okay, if you have group A blood group, and then your antibody will be B, not against your antigens, right? If you are group B and the antibody in your plasma will be anti-A, okay? You shouldn't produce antibody against your antigens, right? If you carry both A and B antigens, okay, there is no antibodies, which is good, right? And type O blood groups will carry, okay, as you see, there is no antigens here, okay? Either A nor B, but there are both anti-A and anti-B antibodies. Now, which help us to understand how we perform in cross-matching. Okay, I'm going to show you. This is a table similar to the previous slides. Now, blood group phenotypes AB, or O, and blood group genotypes, as I said, there are two genotypes, A2 for group B, okay, that can be either both A or A, and which represent group O, okay? But since this is recessive, and both of them is phenotypically group A, okay? Okay, now we have antiserums, right? Anti-A and anti-B, which we prepared before. Now, and we have to test the patient's sample to determine the group of the patients. Okay, and as you see here, okay, you are going to put a anti-sera, anti-A in the test tube and anti-B, okay, in the test tube or in the valve, and then you put 1 drop of the patient's red blood cells on these antiseras, okay? If the patient is group A, what does means the erythrocyte of patients carry A antigen, okay, and then they will react with anti-A, right? And I am correct, agree with me? Type A, okay, and will react with antiserum A, okay, because it's, okay, and then you see here antiserum, the cell containing A antigen will agglutinate with anti-A, right? The cell containing B antigen will agglutinate with anti-B. Okay, the cell contain both antigen, they will agglutinate with both antisera. Okay, if the cell doesn't contain antigens, there is no agglutinations. Okay, now here you will understand better. Okay, type A, cell containing A antigen, agglutinate with the blue antiserum, which contain anti-A. Agglutination causes the cells to fall out to the blue solution. Type B, Okay, cells contain B antigen, agglutinate with anti-B, which is yellow. Okay, type AB will agglutinate with both of antisera, and type O, there is no antigen, no agglutination with this antisera. And then you are going to obtain the results and says to the patients, your blood group is group A, B, AB, or group AO. Now, okay. I am going to show you these videos, and then let's see whether the sound is shared. Okay, now.
In this video, I'm going to show you how to do a forward and reverse ABO-RH type. First thing you're going to need is for your forward, you're going to need your antisera, A, B, and your anti-D. You're also going to need, for your reverse typing, you're going to need your reagent red cells, A1 and B. And then you're also going to need some tubes, and some plasma, and some red cells. Now these are red cells that have been spun, so I need to make a suspension, and I'm shooting for approximately 3%, and I'm using physiological saline for this. If you're not exactly sure what 3% should look like, you can use your reagent red cells to make a comparison. Just give it a good mix. And the reagent red cells should be about 2 to 4%, so if they look the same, you're pretty close. Alright. Once you have all your stuff together, you can go ahead and you can start to label all of your tubes. And then from there, we're going to start with the 4. We're going to add 1 drop of all of these reagents, so 1 drop of each antisera into the appropriate tube, so that was the A. Then you add the anti-B. And then lastly for the forward is the anti-D antisera. Remember, just 1 drop for each of these. And then moving on to the reverse portion, you're going to use the reagent red cells, and once again it's going to be 1 drop of each one of these in the corresponding tube. And just make sure you mix them up a little bit because they tend to settle, the red cells, at the bottom of these reagents. As you'll see here with this, the B, so you can see the cells at the bottom. So just give it a little mix until the cells are gone off the bottom there. And then just 1 drop. Then from here, we're going to go ahead and, I guess you could start either way with these, but I go ahead and start with the plasma. And for plasma, you're going to add 2 drops of patient plasma to each one of the reverse tubes, or the reagent red cells. And then with the patient red blood cells, it's just 1 drop in each of the antisera tubes. And after you've added your patient specimens to each of the tubes, you go ahead and you centrifuge this, and then we'll move on to the agglutination viewer. And what you're basically trying to do here is to gently decant the cell button and just remove it from the bottom of that tube. You can see there we're getting a 0 reaction. All of the red blood cells from the cell button are completely coming off. There's no agglutinations. Again with the B, this is the forward that we're doing here. Once again, we're not getting any agglutination when we decant the cell button or resuspend it. You can see there's a 0 in there. Sorry if this is a little awkward to see. And then moving on to the anti-D. Now this is the first one we're going to get a positive with. You'll see that this has a 4+, really good cell button agglutination right there. And from that forward, you know this is an O+ patient. And then with the reverse, both the A1 and the B reagent red cells are both 4 positive. You can see that single cell button. And that's basically it. You just want to make sure you don't shake these too hard. And there you go. Hope you enjoyed the video.
Okay. So let's see how many slides do we have. Sorry. Okay, let's go ahead for I'm not sure whether we will be able to finish today. Okay, what does universal donors recipient means? I'm sure some of you already know that. Okay, the term universal donor or universal recipient is about the transfusion of packed red blood cell products in emergency situation. Okay, it is only about the packed red blood products and in the emergency situation. Okay, if you know that universal donor is able to donate its red blood cells to the rest of the blood groups, okay, and the universal recipients is able to receive the red blood cells of the other groups. Okay, now the universal donor group is group O, okay, which which doesn't include antigens, right? And universal recipient group B, which doesn't recipient which don't contain the antibodies. Okay, now the other blood group system is RH, red blood group systems, is different from ABO. And the definition for in RH antigen and inheritance is important. There are 5 antigens, D, C, and here you see upper case, lowercase c, uppercase e, and lowercase e, and their corresponding antibodies are of primary importance of routine blood bank testing. And of these 5, the D antigen is the most important one. The presence or absence of the D antigens identify a person cells as being Rh positive or Rh negative. If the antigen is present, okay, we says Rh positive. Okay, if absence, okay, it will be Rh negative blood group. Okay, what that means we are testing both for ABO blood group and for Rh blood group before we transfuse the blood or blood component to the patients. Okay, the types of Rh typing reagents that we are using, okay, high protein antisera, low protein antisera, chemically modified, monoclonal. There are different types of the antisera that we are using to detect the antigens. Okay, when red blood cells are to be transfused to the patients, must be tested for the presence or absence of the antigens. Otherwise, they will react with the patient's antibodies. Now, there are more than 30 different blood group systems, which defined by International Society of the Blood Transfusions. But the most important groups that we already discussed, and we have to know group A, ABO system and RH group blood group system. That should be enough at this level. Okay, the anti-human globulin reaction or Coombs test. This is other test that we're performing in the blood banking. There are two types of the procedure. Direct anti-human globulin test or indirect anti-human globulin test. In the direct one, we performed on the red blood cells suspected of being coated with the antibodies. Okay? Use it to demonstrate antibody that has coated or reacted with the red blood cells of the patients, which is in vivo. Okay? In the indirect, use it to detect antigen-antibody reaction that occurs in the test tubes. I am going to show you each of them. Now, as you see here, if you think that if you do think that the patient's red blood cells synthesized via antibodies, okay, there is a reaction between donor and recipient bloods, and then you have to test them. You have to confirm them. Now, in the direct Coombs test, as we see here, you are withdraw a blood sample of the patients, okay, and if they are, this is in this case, this is a immune-mediated hemolytic anemia. What does means there are a reaction between erythrocytes and antibodies. Okay, and in this case, if you withdraw a blood sample of the immune-mediated hemolytic anemia, there are a reaction between antigen on the surface of the red blood cells and antibodies. Okay, these antibodies may came from the donors. Okay, now you withdraw the blood and then add a anti-human globulin antibodies. Okay, these are antibodies, anticorps. Okay, now, and they will attach. These are specific to the antibodies, which already react with the erythrocytes, and then they will attach to FC portion of the antibodies, the clumping. Okay, and then they make agglutination visible. Now, in the indirect Coombs test, the recipient serum is obtained, okay, which serum contain antibody, right? And then the donor blood sample is added to the tube. Recipient immunoglobulins, which the donors' bloods contain erythrocyte, okay? Now you have recipient serum and donor red blood cells. If there is antibodies in the serum, they will attach, and these antibodies specific to the antigens on the surface of these erythrocytes, they will attach them, okay? However, you may don't see the agglutination visibly, and you add anti-human globulins, Coombs antibodies, in order to make the reaction more visible, okay? Now, agglutination is visible with your naked eyes, okay? Okay, the compatibility testing and cross-matching are really important. If you make a mistake, you may kill the patients, okay? And therefore, whenever red blood cells are to be transfused, two considerations need to be confirmed. Okay, these are important, and the red blood cells must be selected that will not be harmful to the patients or result in a transfusion reaction. Red blood cells must be selected that will be of maximum benefit to the patients. Okay, adverse effect of the transfusions, which we scare of them. Therefore, we are really pay attention. We perform the cross-matching test very carefully in order to avoid such the transfusion reactions. Okay, clinically, the result of the red blood cell restrictions is known as transfusion reactions. If you transfer a blood which carry antibody to the erythrocytes of the recipients, then they will react, and then they will make a restriction of the red blood cells. The sign and the symptoms of a transfusion reaction vary from patients to patients, is based on the severity of the restriction. Generally, chills, high temperature, pain in the lower back, nausea, vomiting, shock, which is an indication of decreased blood pressure and rapid pulse. And these are the general symptoms you may see, which characterize the immediate reactions. Okay. Okay, these are the most common adverse effect of the transfusion: immediate immunologic adverse reactions, such as transfusion-related acute lung injury, we call TRALI, hemolytic transfusion reactions. It can be either acute hemolytic transfusion or delayed hemolytic transfusion, or immediate non-immunologic adverse reactions, and other severe adverse reactions may be seen as an adverse effect of transfusion.